Hello everybody and welcome back. Today we're going to be talking about the myocardial cell structure. The myocardial cell structure is an involuntary striated muscle which contracts in a synchronous fashion. They all contract in the same time. The cardiac muscle cells have a diameter of 10 to 20 micrometers. They also have a length of 50 to 100 micrometers in length. The structure of the myocardial cell has different components. The sarcolemma, the first component, which has T-tubules and terminal cisternia, and also the sarcoplasmic reticulum, which forms the excitation-contraction coupling. The second structure is going to be about the sarcomere. The sarcomere is a contractile unit aligned across the cell, which gives the striated appearance of the myocardial cell. It has myofibril proteins. The contractile units, which are the myosin and the actin, and the regulatory units, which are the tropomyosin and troponin. It also has accessory and non-contractile cytoskeletal filaments like tropomodulin, titin, connectin, and nebulin. For the third structure in the myocardial cell is the intercalated discs. The intercalated discs are mechanical connections like desmosomes and electrical junctions like gap junctions, which form a syncytium. During an action potential, the entire heart has about 220 milliseconds of contraction in the myocardial cell. In the sarcoplasm of the myocardial cell, it contains myoglobin, which is about 3.4 grams per liter, and oxygen storage, which is 50% saturated at PO2 of 5 mm mercury, which facilitates the diffusion of oxygen through the sarcoplasm. The myocardial cell structure also has a single central nucleus. It also has a mitochondria, which reaches up to 30% of the volume of the heart, which causes a great oxidative capacity. The myocardial cell structure also contains rich capillary supply. One capillary supply has one myocardial cell, so for each cell it has one capillary supply, and short diffusion distances too. So for the sarcolemma, we're going to start explaining by the sarcolemma in details. The sarcolemma contains the transverse T-tubules and the sarcoplasmic reticulum. For the transverse T-tubules, they contain invaginations of the sarcolemma with the extension of the extracellular fluid. The transverse T-tubules are also more developed in the ventricles. They have a scanty in atrials and Purkinje cells. They are also oriented at the Z-lines, and they enable fast impulse transmission, almost simultaneously with stimulation of myofibrils. So these were the T-tubules. For the sarcoplasmic reticulum, it develops from the endoplasmic reticulum. It stores calcium storages. It's closed set of an anastomosing tubules wandering through the myofibrils, like the network of the sarcoplasmic reticulum, which causes calcium reuptake at calcium ATP pumps and phospholambane. It also has junctional sarcoplasmic reticulum, which is close to the sarcolemma and T-tubules and also stores calcium. And for the corbular sarcoplasmic reticulum, sac-like expansions, which are along the sarcoplasmic network in the I band. They also store calcium and calciquestrine. Now with the sarcomere. The sarcomere is the basic contractile unit of the myocardial cell. It's located between two Z lines. It has about 1.8 to 2 micrometer length in resting myocytes. The Z lines are thin, dark staining partition components of alpha-actinin. The actin is thin, 6 nanometer. Filaments extend from the Z lines toward the center of the sarcomere and overlap the myosin. The myosin, which is the thick filament. So actin is thin filaments, about 6 nanometer, and the myosin are thick filaments, about 11 nanometer filaments. The actin filaments form the pale I band, I from the isotropic, and the myosin filaments lie in parallel in the central region of the sarcomere called the A band, the A from anisotropic. Each myosin is surrounded by 6 thin filaments forming a hexagonal array. The major components of the cardiac sarcomere are myosin, which form actin interactions with ATP's activity, the actin, which have ATP and calcium and magnesium binding sites, which form interaction with myosin and troponin and tropomyosin. The contraction is the shortening of the sarcomeres. The I band shortens, not the A band. So for the contraction, only the I bands shorten. And sliding the filament mechanism, which is repeated making and breaking of cross bridges between the A and M filaments, the actin and the myosin. The cross bridges are the heads of myosin molecules, which change their angles by binding to the actin sites. After tropomyosin, calcium-dependent displacement. So these were the myocardial cell structure. Thank you very much and hope to see you soon again in the next video.